In 1880, rainbow trout native to the Pacific coast of North America were shipped to Germany as a food fish. They spent the next century in Bavarian hatcheries co-evolving with a parasite that didn't exist in American waters. When the parasite arrived in the United States and began collapsing wild trout populations, the solution was already in Germany, inside the descendants of American fish. Myxobolus cerebralis was accidentally introduced to the United States in 1957 through infected fish. When it reached Colorado in the late 1980s, it spread to 40 stocking locations within a decade. Wild rainbow trout populations fell to 10% of historical biomass. By 1997, 10 of Colorado's 14 state hatcheries were infected, and natural reproduction had stopped across much of the state. At a national conference in Denver in 2002, German researcher Mansour El-Matbouli presented findings on a strain of rainbow trout at the Hofer Trout Farm in Bavaria, showing markedly reduced susceptibility to the parasite. George Schisler of Colorado Parks and Wildlife, working with UC Davis, imported Hofer eggs and tested the fingerlings. They were 100 times more resistant to whirling disease than Colorado's own strains. Genome expression studies identified part of the mechanism. After parasite exposure, a gene called metallothionein was unpregulated over 5-fold in Hofer fish and was unchanged in susceptible fish. The Hofer fish had a critical limitation. Raised in hatcheries for over a century, they'd lost their predator avoidance behavior entirely. The solution was crossbreeding, combining Hofer resistance with Colorado wild strains to produce fish with both the immune architecture and the behavioral capacity to survive in rivers. The resistant allele maps to a quantitative trait locus called WDRES9 on chromosome OMY9. A 2025 paper from Colorado Parks and Wildlife and UC Davis, published in Frontiers in Freshwater Science, is now using DNA-based field screenings with the WDRES-9 test to identify which Colorado fish naturally carry the resistant allele, allowing managers to protect these individuals specifically. Fish exported from North America in 1880 as food came back 120 years later as the genetic foundation of a conservation program trying to save the species they came from.